to all our esteemed guests, respected faculty member, dear students, and valued member of the Microbiology Society. I extend a warm welcome and lecture. I'm Shweta Phattare, honored to be the convener and your host from this session. We are delighted to connect with all of the virtual platform for an evening scientific learning and meaningful insight. Before we begin, I kindly request everyone to keep their microphones mute, ensure a experience. The Microbiology Society continues to serve as a dynamic platform for promoting education, research, and collaboration in the field of microbiology. Under the leadership of our president, Dr. A.M. Deshmukh, sir, we have been organizing impactful programs that encourage innovation, critical thinking, and scientific excellence. Thank you for being a part of today's session. It is my privilege to introduce today's guest speaker, Dr. Surendra Vikram, sir, a globally recognized microbiologist, computational microbiologist, and expert in microbial ecology. Dr. Vikram, sir, is currently serving as an assistant professor in the Department of Biotechnology at Maharshi Mahesh Yogi Ramayan Vishwavidyalaya, Ayodhya, and is also the founder of Bio Private Limited, Ayodhya. His academic and research journey is exceptionally rich, spanning India, South Africa, and global research collaboration. Completed his PhD at the CSIR Institute of Microbial Technology, Chandigarh, with affiliation to Jawaharlal Nehru University, New Delhi, where he carried out groundbreaking genomic study on Burkholderia species. He has worked extensively as a postdoctoral research fellow at the Central for Microbial Ecology and Genomic University of South Africa, contributing to high-level projects involving genomic, metagenomic, acid and DNA analysis, and microbial ecology of extreme environments such as the Antarctica. His expertise include next generation sequencing, bioinformatics pipelines, genome assembly, metagenome assembly genomes, microbial community analysis, and computational strategy in microbial ecology. Dr. Vikram sir has authored numerous high impact research publications in international journals such as Scientific Reports, Fungal Biology, Microbes, PNAS, Environmental Microbiology, and Frontiers in Microbiology. His research extends across diverse areas including soil microbiomes, Antarctica microbial communities, acid and pathogen genomics, and next generation sequencing analytics. He has been awarded several fellowships, including the CSIR GRF, CSIR SRF, and the Cloud Leon Foundation postdoctoral fellowship in South Africa. He is also served as reviewer and associate editor for international journals, reflecting his deep expertise and global recognition in microbial bioinformatics. Today, he will be speaking on a highly relevant and rapidly advanced topic, decoding the microbiome, computational tools, and bioinformatics strategy. This session will give us an opportunity to understand how computational biology is transforming our ability to study microbial communities, explore hidden diversity, and unlock functional insight from complex sequencing dataset. Without further delay, without further delay, I honored to invite Dr. Surendra Vikram sir to take over and share his knowledge, experience, and scientific with us, sir. The virtual stage is yours.
Thank you very much, Ms. Sweta, ma'am. And I would like to thank you, Dr. Deshmukh, sir, for giving me the opportunity to present here in this forum here, and which is a very highly reputed forum, which is connecting the scientists all over the world, and including the Indian very renowned scientists. I would like to thank you also the MBSI uh, group for giving me this opportunity. Right, and then so now I will move to the topic here, which I have selected, which is so I'm not going to discuss today here what I have done in the past, but more over the focus will be on how we can perform the microbiome studies. So I will give some of the tools which are being used in the microbiome analysis, and then the basics of microbiomes, like including the metagenomics, and then the parts of the metagenomic studies. Right now, am I audible? Please, I think
Yes, we can hear you, but somebody is unmute.
yes,
We can hear you.
right. So this is the very basic slide which I have prepared here to tell or to show us that we were not knowing how the microorganisms are behaving in the environment. These pioneers in the microbiology, they have discovered the very small organisms which cannot be seen by the eyes. So this is the father of microbiology, which is Anthony van Leeuwenhoek, which has discovered the animalcules, and then later on they were classified as the microorganisms or the bacteria. Then the Louis Pasteur, which is known as the father of the modern microbiology, which has identified and then put the foundation for the pasteurization and then the techniques which are used in the food microbiology. Then the Ferdinand Kuhn and the Robert Koch. Robert Koch is also one of the father of the medical microbiology, which is known as, which isolated different types of the bacteria along with the anthrax, tuberculosis, and cholera. And then he also gives the Koch's postulate, where the organisms can be the causative agent of the diseases can be transferred from one organism or from one to the any other organism. So the diseases can be transferred by the transferring the positive agent. So that was the sort of the Koch postulate which was given by the Robert Koch. So these were the pioneering scientists or the persons which have started the field of microbiology, and they have given a lot of insights from their understandings what they have studied there. So this is not limited to these only like four faces which I'm seeing, but this is like there is a whole list of the people which were involved. But I'm not going to include all of them, but to show here that these are the most important persons in the field of microbiology. Then when we talk about the molecular biology and how we can go from understanding and studying the microbiology or the microorganisms, so most important aspect is to first to identify the content of their genome or how they are different from one another. So like the sequencing technology revolution, like the identification of the Thermus aquaticus, this was discovered by Thomas de Brock, and then later on Carey Mullis invented the technique of polymerase chain reaction, which and then the later on there was the identification like the foundation of the next generation sequencing platform. So that was the Jonathan Rothberg, who has founded the 454 Life Sciences, and then this initiation of the high throughput sequencing. So the DNA was sequenced on the high throughput sequencing platforms to produce like millions or billions of the copies of the template DNA, which was being transferred there. So like in the pipeline, like 454 sequencing platform, and then later on the Illumina, which is dominating the field of sequencing technology right now, like the Illumina MiSeq and then the Illumina HiSeq, they were introduced in different era, like in the years. So this is a very highly efficient technology where we see the Illumina MiSeq and HiSeq, they generate like a huge amount of data they generate here, and then there is like the very low error rates which we identify in the sequences. So like they claim here that there is only 0.01% of the error rates, which is a very high percentage of the correct sequences we can obtain from the Illumina sequencing platform. Right now, okay, right here. Sorry, pointer I want to use here, but it is not working. Right. So the third generation sequencing platform, which has been which is in the market, which we see here, that the Bio and the nanopore. So these technologies, they generate a lot of longer sequences. Like in the limitation of the Illumina sequencing platform is that we can get only up to 300 base pairs from one end. So if we sequence it from both the sides, forward and reverse primers, we can cover 600 base pairs of the template DNA. But when we talk about the next third generation sequencing, like the Bio or the nanopore sequencing technology, so it can cover up to 50 kb of the sequencing, or the 50 kb of the template DNA to generate the sequences. But the drawback of this third generation sequencing platform is like they have the higher error rate. So they have like up to 12% of the error rate, which is quite high. And then the reliability of the data is questionable. They are keep on working to improve the quality of the data, but it is having the higher error rate. So if we combine these two technologies, we can get a good output. So I'm not going to discuss more about this, but for I will go to the microbial ecology side here. So this is the background which I gave you here for the sequencing technologies that is very relevant in the field of microbial ecology. So when we talk about the ecology as a whole, so the ecology stands for the study of the organisms in an ecosystem and their interaction with the other biotic and then the abiotic factors like pH, temperature, and then how the different environmental conditions are affecting the organisms and how they are behaving in that particular environment. The same way, when we talk about the microbial ecology, so the microbial ecology stands with the microbiology of the microorganisms, how they are relating with the other organisms, how they are interacting with other organisms in an environment. Like, so what are the effects like the abiotic and biotic factors are posing on that particular environment? So when we talk about here, the microorganisms, so this is the scale where it starts from 0.1 nanometer to 1 kilometer. But we see here, when we talk about the microbial ecology, which is ranges here up to from viruses, if we want to include the viruses also, it is like 100 nanometer or 50 nanometer to the up to 600 to 700 micrometers. So they are like the micro eukaryotes, then the fungi is also there, nematodes are there, then the microorganisms in the case of bacteria and the archaea also, they are included. So that is the in a nutshell, when we study the microbial ecology, we focus on the organism which cannot be visualized or identified by the naked eyes. Right. So when we talk about why it is important, like the microorganisms are important, but why they are important? Because they dominate the tree of life. So when we talk about how they dominate, so the in the background, when we talk about the Carl Woos and the colleagues, they have identified the SSU gene, that is a small subunit rRNA gene, which was used for the construction of the phylogenetic tree, so which has revolutionized the classification of the organisms, where it was identified that we can classify the organisms into the three different groups like bacteria, archaea, and the eukarya based on the phylogenetic analysis of the small subunit RNA. In the case of bacteria, it is 16S rRNA, and in the case of eukaryotes, it is the 18S rRNA. So based on that, phylogenetic tree was prepared, and then it was classified as the bacteria, archaea, and the eukarya into three domains. Right. So on the right side here in this figure, what we are seeing that the red dots which are shown here, they are the unculturable diversity. So we see here, this is the tree of life, which was published in 2016 by Hug et al. In Nature Microbiology. And they have identified like lot of different or new species of the bacteria. So we see here a lot of different new species were identified. When we talk about these italic names which are written here, they are the culturable organisms or the representative of the culturable organisms here in the tree. So it means that there are a huge number of the bacterial diversity which can't be culturable. Until date, or still, we don't have the ability to culture the organisms which are like more than 99% of the organisms are unculturable. So what are the reasons, what are the problems there to culture them? So there are various different issues related to the culturability of the organisms. the most foremost reason is that we cannot define the particular conditions to grow the organisms in our laboratory. So that is the one issue what we when we face the problems to culture the organisms. So which can be overcome by studying the microbial ecology, their interaction with the environments, and then we can identify what conditions will be suitable to culture the organism. So that is like a reverse metagenomics or the reverse genomics studies. Right. So if this is the high diversity which is being unculturable, how to study that? So in the field of microbial ecology, we can implement various different techniques to study. So here we see the central dogma where the DNA is transcribed into RNA, and then RNA is translated into the protein. So we can utilize these particular central dogma pathway like the DNA, RNA, and proteins. We can use them to study the microbial ecology or the microbial diversity of particular environment. So whatever the environments we can select here, like suppose we want to select food samples, we can select, or we can select the soil samples, and these samples can be obtained from any environment, like from the desert environment or from the forest soil, from the agriculture soil. So from where we can select the samples, and then we can perform these particular techniques on the to study the microbial ecology based on the unculturable techniques, because there is like more than 99% of the organisms which are unculturable. So when we talk about the DNA, we extract the total DNA or the metagenomic DNA. So that is called the metagenomic DNA from the total sample. We extract it and we study. So there are two methods we can apply here. One is the amplicon sequencing, another is the shotgun sequencing method. So when we talk about the RNA, so we can also extract the RNA template or the mRNA, and then we can also sequence it. So we extract the total mRNA from the environmental sample and then perform the meta-transcriptomic study. So we can identify the microbial function there, how the RNA is being transcribed, and then we can identify those particular RNA sequences. We can relate the function of those mRNA by aligning to the reference databases. The other method is the protein extraction. So using the protein molecules by extracting it from the environmental samples directly, and then we perform the metaproteomic studies. So in the proteomic study, we can use the mass spectrophotometer based, like the MS studies, or the technique can be utilized to identify the protein sequences there. Right, so this is the microbial function. When we use the RNA and the protein based studies, we can understand the function of the microbial community which is present in any environment. We can also extract the metabolites from any environmental sample. So the metabolites which are being extracted, it means if we find particular carbohydrates or particular different types of the molecules which are present there, so that is they are the product of the enzymatic conversion, or they are the product from any of the metabolic pathways. So it means that they are the real activity which is present there. So this is also the activity of the microbes which are present in any environment we can identify there. So when we talk about DNA, RNA, and protein, we still study only the microbial potential here, because we cannot be sure that whether it is present, the RNA is present, but if it is functional or not, we are not sure. Then the same way it happens with the proteins and then the DNA. So we only identify the potential of the microbial community or in any environment. Right. When we involve all these techniques here, like the multiple omics techniques, multi-omics techniques like metagenomics, metatranscriptomics, metaproteomics, and metabolomics, we perform the multi-omics or the system biology approach, where we apply the mathematical models and then the statistical models to identify the total functions or the total potential and then the activity of the communities in any environment. And that may lead to the study of the climate change, and then the modeling the climate change, desertification based on the differences in the community, and then the environmental factors we can implement there. So that is like a whole picture can be generated by applying all these techniques together there. So when we talk about the microbial community, when we see here, this is the like the hierarchy of the microbial community in any ecosystem. So when we talk about here, like the individual organism or the individual microbe, so that is like a single cell. So when there are multiple organisms are there or a microbe is there, then that is called the population, but they are the same species. The guild is like when there are one or two species or more species are working to compete in the same environment. So they form the guild here, microbial guild in that particular environment. So they compete for the resources in the same environment. The community is like when there are multiple guilds, they are present in any environment. So they form the community, or we can say here in the case of microorganisms, they are called the microbial community. And then the ecosystem is when they are interacting with the other organisms. Suppose in the case of gut microbiota, so they are interacting with the host and also with the other biotic and the abiotic factors. So that is the ecosystem where we see here, like in any ecosystem, how those microbial communities are behaving. So when we want to study the microbiomes or the microbial ecology, in that case, we select the samples. So first, the sampling is very important in that case. So the sampling strategies are there, like biological replicates. We need to select the samples there, right? We collect the samples, but there should be replicate samples. So there are two types of the samples can be selected, like the biological replicates and the technical replicates, depending on the question we pose and what we are going to identify. So in the case of here biological replicates, the independent samples that reflect the inherent variability of biological condition or the population being studied. When the population is being studied, what are the biological differences between the organism or within the species which are present there? So that can be analyzed. Right. So that is why the biological replicates are important. Suppose we have selected multiple organisms in a sample group. So the replicates are important to identify whether there are differences in the same organisms, or that is the effect of something what we are going to study, like the effect of drugs or some molecules, or the season, like different conditions like pH or temperature. So whether to find the differences, we have to put the biological replicates to discriminate between the biological conditions and then the effect of that particular condition which we are going to study. So the examples of the biological replicates is like samples taken from different individuals in the population. So like the cancer patients, but there will be replicates. So the same way, the control samples, but there will be replicates. The different plants are grown under the same condition in an experiment. So we can also select the different plants which are grown in the same condition, but replicates. Cultures of the same microbial species grown from different colonies. So that can also be selected. That will be selected as a biological replicate. When we talk about the technical replicate, which measures or analyzes the same biological sample to account for variability introduced by experimental procedures, like the pipetting error or the different types of the instrumentation errors when we do the sequencing technology. So whether the sequencing was done properly or it was the library preparation was performed well. So that is the case of the technical replicates. So it poses like depend on the experiment we are performing. If we are studying the proper microbial ecology, then it is important. The biological replicates are important to include. When we are doing the technical replicates, like we are testing or preparing any protocol or something to SOP, we are writing any SOP to perform some experiments. So the technical replicates become more important. So running the same DNA extraction on one sample multiple times, performing PCRs from the same cDNA. And the repeating sequencing runs of the same library in the sequencing technology. Right, so this is the part which I have covered here for the sampling strategy using the biological replicates and then the technical replicates. Now I will come to the metagenomics part, which I'm going to cover in the detail here. So when we talk about the metagenomics, it is separated into two forms, like the amplicon sequencing, data analysis, or the amplicon sequencing, and then the other one is the shotgun sequences, which I have introduced earlier in the previous slide. So we can select any of the samples from the environment, like soil, water, clinical sample, or the food sample. What we do, we isolate the DNA. Total DNA is extracted, and then this DNA is used for the amplification of particular marker gene. So in the case of prokaryotes, we select here 16S rRNA gene. This gene is very important because it has the conserved region and also the variable region. So conserved regions can be used to prepare the PCR primers, and then the variable regions can be used for the identification of the variation in the species. And this is well preserved, like this particular 16S rRNA gene is evolving together with the speciation. So there is very little change happens in the during the replication or the division of the cells. It is preserved. So when the speciation happens, then the 16S gene also that get changed, that is evolving there with the different species. So the PCR primers are used to amplify the 16S rRNA gene here. Then there is the high throughput sequencing of the amplified genes are performed on any of the sequencing platforms. So the Illumina sequencing platform is very popular to do the high throughput sequencing here. Once the sequences are generated, they are in the short reads. So we get the sequences in the length of 300 to 500 base pair length. Then we use the bioinformatic tools here, like the Chime2 and R packages. So R is a software, and the packages which are available there, they are the they are called the R packages. So we can implement these two pipelines, or there are other tools also available. I will discuss later in the presentation. So these are the pipelines, these are the tools that can be used for the identification of the clusters in the data. So this is the amplification which will be amplifying the 16S depending on the template DNA. So if there are two template sequences for particular species present, so that will amplify it in the exponential number. So the same way, if there are 10 species were present or 10 bacteria were present for that particular species, so that will be amplified in that ratio. So by that those sequences are clustered, and then the counting of those sequences are done, how many times these sequences were present there. After that, we can identify the taxonomic classification there. So based on the database search, like the Silva or the Green Genes database for the 16S rRNA gene is used for the classification of these sequences to assign the taxonomic lineages, like the kingdom, phylum, class, order, family, genus, and species level. The other way we can also perform here, like based on this clustering, we can perform the phylogenetic tree, phylogenetic based analysis. So what we do, we align the sequences and then we create the phylogenetic tree, and then the further we can study the diversity of these organisms. Once the taxonomic classification is done, we can perform the bar plot, or we can identify which species is present in this particular sample higher, or in this particular sample which is lower. So that is the comparative study we can perform here, like which species was important in this particular environment and which was not important in this on the other environment. Or the same way we can also use the clinical sample, or the treated samples with the drugs or the fertilizers in the agriculture. So these are the important aspects to study here, like what is the effect of the different questions which we are asking there. So how the microbial community is behaving in certain environment, in certain conditions. So by studying the amplicon data here, like the marker-based sequences, we identify the microbial taxonomy and the diversity, but we cannot identify the functions and the metabolic information of the community which is present there. In certain cases we can do, I will discuss that also later here. So when we have the the amplicon data, and then we have the phylogenetic analysis here, the diversity based approach. So what we can do here, we can assess the diversity indices, alpha, beta, and gamma diversity indices can be assessed here for the microbial community. When we talk about the alpha diversity, this is the biodiversity within a particular area. That is the number of species in that area, that all, or also it is the species richness. So it, suppose there are, like, we have 3 sites and we have 10 species present in site A, 20 in site B, and then the 5 at site C. So that is the alpha diversity, like 5, 20, and 10. So that is the alpha diversity. The beta diversity is the comparison between the different sites or the area. So when we compare the 5 microorganisms from one side to the other side, that is the beta diversity. So differences between the sites, that is called the beta diversity in the community. Then the gamma diversity is, it covers the whole diversity of, including the alpha and beta diversity in the particular area. So how we can identify the different number of the species which are present in an ecosystem, that is called the gamma diversity. This is just a very basic example what I'm showing here. Like at the site A, alpha diversity is 3 because there are 3 different species present here. At site B, the alpha diversity is 3 with the presence of 3 different species. At site C, alpha diversity is 2. This is the species richness. When we talk about the beta diversity, so the difference between the site A and B, so it can be compared with the site A and site B. Here is 3 species, so 3, and here is 2 are present as the unique here. They are the common. Right here, 1 is unique and 2 are common. So 3 minus 2 between A and B. And then there is 3 and 2 are common here again. So 3 minus 2. So the beta diversity between A and B is 2. The same way beta diversity between A and C sites is 3. Like 3 are here present, 3 species are present, and then 1 is common. So 3 minus 1 and then plus 2 minus 1. 2 is present here and then 1 is common in between them. So that is the beta diversity between A and C is 3. The same way beta diversity between A and B and then B to C can also be calculated here. When we talk about the gamma diversity, here the gamma diversity of this ecosystem is 5 because there are 5 different species are present, right? So that is the gamma diversity of the ecosystem, which is at these 3 sites, which is 5 different species. And these are very important indices which are used in the microbial ecology to understand the differences between the sites or between the treatment groups. We can identify there. So if there are like different species present, we can say that the treatment is affecting the microbial community. If that is lower, if it is higher, then we can say, okay, the treatment is enhancing the microbial communities and then it is supporting their growth in that particular environment. Amplicon or marker-based metagenomics can be used here. So this is just for the summary slide of the amplicon-based here. Like what we can do by using the marker-based metagenomics or the amplicon metagenomics. What we can do, we can identify the taxa which is present in a community. We can highlight the dominant and the rare taxa. So what are highly present there and then which are very rarely present. So that is also the important aspect to study. Then there is the diversity analysis we can perform. Alpha diversity and beta diversity studies between the sites we can study, and we can identify where there is the difference which is significant or not based on the statistical analysis which we apply there. Functional interference and biomarker discovery. So this is the process which we can perform after having the sequences from the marker genes or the 16S sequencing output. But this has the limitation because it is using the 16S genes to identify the functions, and which is not very confident. It is only identifying that this particular bacteria is present in this environment. So this function might be present there. So that which is not very confident and not very accurate. But still, the scientist and the community is using these tools like PyTrust and Tax for Fun to identify the functions based on the marker genes. Then there is a temporal and spatial dynamics studies can be performed. Like we can use the time dependent studies, like we sample at different time points and we also sample at different distances, and we can study the differences in the microbial communities. So that is like, this is compared to the shotgun sequencing or shotgun metagenomic sequencing technology, this marker based sequencing is cheaper. So that can be used for these type of the studies, like temporal and the spatial dynamics studies. What are the different tools which can be used for the amplicon sequencing data analysis? They are like OTU or ASV or ZOTU construction. So I will explain it later in more detail, little bit about what are the OTUs and the ASVs and ZOTUs. Here we see the mother, like a tool which can be used for the construction of the OTU table. Like we can cluster the sequences based on their differences or similarity. The same way we can use here the usearch tool for the ZOTU construction. Data2 and Dibbler, another tool which is not listed here, they can be used for the ASV construction. Chime2 pipeline is used for the analysis by using Data2 and Dibbler2. So these are the different tools which can be used for the study of amplicon sequencing data and then the analysis of that particular data which we have generated here. Then there is the downstream analysis we can perform also, like Chime2 pipeline. It includes various different modules to perform the analysis. And I also introduce the term here R packages. So R is a software, it is a programming language where the various different packages, R packages available. So these were developed by the scientific community where we can use these different packages to perform the microbial ecology studies by performing the alpha beta diversity, and we can also perform the multivariate analysis while we can do the PCA plots and PCOA plots we can generate there. So this is the basic uh, structure or basic representation of the OTU or ASV table. So here we see on the samples on the column names. So these are the samples. Suppose this is the sample 1, sample 2, sample 3, and this is the OTU IDs on the row names here. These are the OTU IDs. And we see here, this is the frequency of these OTUs. So this is the number of sequences they were identified in each sample. So here we see OTU6 is present 749. The same way here in F3D147 sample has 2025. So this is the OTU table which can be used for the downstream analysis for plotting the graph for the taxonomic analysis, and we can also perform the various alpha and beta diversity analysis. When when I talked about the OTU table and the ASV or ZOTUs. So operational taxonomic units are the most common approach to group the sequences that are more than a certain percentage. So if they are showing the 97% similarity, they will be grouped as a one cluster. If they are showing less than 97% similarity, those sequences will be clustered as the different OTU. So that is how the OTUs were generated for this particular table. The same way ASVs are generated, but the concept is different. I will explain that very soon. So what are the limitations of the OTU table? So these OTU tables are limited by the chosen similarity threshold selected for the OTU construction. But still it is used, which is not very confident. And or underestimation of biodiversity, primarily when sequences differ by only a small amount. So the if the sequences have very small changes in there, then it is also not very informative, right? So this is also sensitive to the sequencing error or biases there. When we talk about the amplicon sequence variants, so that is the more refined approach, and which is the more recent concept and very widely used. So OTUs are not very widely used, like they are being discarded day by day. Now the ASVs, like the amplicon sequence variants or the zero radius OTUs, are being implemented, or they are used. They are more refined approach compared to OTUs. ASVs are unique sequences identified directly from raw sequencing data without clustering. And they represent a distinctive variant of amplicons based on its exact nucleotide composition. So if there are like one or two nucleotide differences, they will be clustered as a different ASV. So that the table will be created the same way, but they will be called the ASV table or the ZOTU table. ZOTU is the zero radius OTUs or operational taxonomic units. Right. So what are the limitations of the ZOTUs? That they are very sensitive, but if the errors are there in the sequencing output, if the sequencing output is not very healthy, then there will be a problem in classifying the ASVs or the ZOTUs. This is a pipeline of the chain 2, which uses the OTU construction and then the phylogenetic analysis and the taxonomic analysis. So, is it fine? I should go with this, or I should move on because I'm running out of time. Is it okay, sir, to take 15 minutes more?
Yeah, you can explain.
Okay, sir. So the raw sequences are used here, which are generated from the sequencing platform. And then the demultiplexing means if the sequences were multiplexed there, they will be demultiplexed. Like during the single run, there were like multiple samples used. Then the demultiplexing is done based on the barcodes, which were used during the multiplex sequencing. So demultiplexing is to assign the sequences back to the samples, each sample, based on the certain length of the barcodes, like 8 to 12 nucleotides long barcodes are used here. Then these demultiplex sequences used for the denoising and clustering. So the denoising and clustering is identifying the sequences which are similar and where are the differences. Based on that, they are clustered into separate groups. Then there is the feature table is constructed. So this is the OTU table also. This is a synonym for the OTU table here, feature table, which is constructed. And then the representative sequences for each cluster is selected. So representative sequences can be used here for the taxonomic classification by aligning it to the reference database. And then the alignment can be done and to perform the phylogenetic analysis and then further to the diversity analysis. Here, and then the statistical analysis to perform the alpha beta diversity and then also perform the multivariate analysis. Then the taxonomic classifications can be used to plot the bar plots, heat maps, and then the differential abundance of the different microbial species which are present there to identify in different samples. Right, so this is the pipeline which is the CHIME 2. The same way there are other pipelines also available like the Mother 2, which can be used to perform till here. This particular analysis can be performed here, like following to construct the feature table and then the representative sequences, and then further we can perform the different types of the analysis like the classification of the taxa and then the alignment and then further the phylogenetic analysis. So now I have discussed here for the amplicon sequencing. The shotgun sequencing is different. Like we don't target a particular sequence here. In shotgun sequencing, we extract the DNA from the environmental sample and then we construct the DNA library of total DNA, which is present there. After that, we use these sequences to align to the reference database like the NR protein database or any other reference database to identify the taxa as well as the functions. So genes can be identified here based on the alignment to the sequences to the database. And then further, we can perform the what are the different genes are important. And then we can also construct the metabolic pathways based on the identification of those genes there. So what we identify here, we identify the microbial potential using the shotgun sequencing technology. What further we can do? We can identify the functional processes, metabolic potential, and pathogenic traits by using the shotgun sequencing method here. Like the functional potential or the biochemical cycles like carbon cycle, nitrogen cycle in a particular environment, sulfur cycling, and then the phosphorus cycling can be identified there. The different tools which can be used here, that is like the KEG database can be used here. KEG that stands for the Kyoto Encyclopedia of Genes and Genomes. Gene ontology annotations and the enzyme classification systems can be used to identify the different functional processes in any environmental sample, in any metagenome. Then there is metabolic potential can also be identified, like how the organisms are behaving, what are their metabolite, their enzymatic pathways or the metabolic pathways which are present in any organism. Suppose the carbon and nitrogen metabolism, antibiotic resistance mechanisms, and the biosynthesis of secondary metabolites as the antibiotics, and then also the terpenoids and then other metabolites. So the tools which can be used here, that is the pathway tools. Metaseq is another tool which can be used for the metabolic potential study. Human is for the human gut microbiome study to identify the metabolic potential of the microorganisms in the metagenomes. And then the MAG-based reconstruction, that is the metagenome assembled genome-based reconstruction of the genomes, can also be used to identify the metabolic potential of the organisms in environmental samples. Pathogenic traits of the metagenomic data can also be identified. So there are various different tools available, like this is the VFDB database. There's CARD, Comprehensive Antibiotic Resistance Database, and AMR plus database, which is available to identify the pathogenic traits of the organisms from the environmental samples. The virulence factor database is also, that is VFDB, which is available to explore the metagenomic samples or the metagenomic data to identify the virulence traits for the organisms. The different tools which can be used here for the shotgun sequencing analysis, that is, like, one of them is the Megan 6, which is a very popular tool, which we can go to this particular link here and then download the Megan Community Edition. So this is Community Edition is free for all the academic research. But if we want to use the Megan 6 Ultimate Edition, which has more features, then we have to pay certain fees for the usage. So Megan 6 CE can be used by anyone for the academic purpose. Then there is another tool which is Kraken 2, which is used for the classification of the taxa from the shotgun sequencing reads. So we can use the Kraken 2. Centrifuge is another tool for the classification of the sequencing reads. Metabat 2 Concoct can be used for the reconstruction of the metagenome assembled genomes. So the genome bins can be reconstructed here using the Metabat 2. Virus sorter 2 is the tool to identify the viruses, like bacteriophages and the other viruses in the environmental sample. So this virus sorter 2 can be implemented with the metagenomic data. Downstream analysis can be performed again in the R and R packages, which is a very important tool. Various different tools can be used, or the R packages can be used for the analysis. So when we talk about the shotgun sequencing data analysis, this is just a very basic example of the analysis pipeline where we implement like Diamond plus Megan. So Diamond is a tool which is like 20,000 or 10,000 times faster than the BLAST-X analysis, because there are millions of reads we have to align to the NR database, which is a very huge database. So to perform the BLAST-X here, we can use the Diamond, which is very fast alignment tool, to the NR database. Then we implement the Megan pipeline. We use the Megan pipeline to perform the taxonomic and the functional binning there. So once the Diamond, then the Megan pipeline, and then we can identify the taxonomic affiliations along with the functional annotation. For that, we can also do here the seed annotation. We can perform the enzymatic classification, geo classification, and EGNOC classification we perform here in the Megan analysis. Then the next step which I showed here, like the construction of the metagenome assembled genomes. Why it is important? We can perform the metabolic potential of each and every organism which were unculturable. So we can identify the particular genomes from the metagenome and then reconstruct it and then study the metabolic potential. We can identify the novel pathways, and we can also identify what are their nutritional requirements so that we can culture them in the lab. So this is just the example here. we construct the reads here by extracting the DNA, and then we can generate the assembly by using the assembly tools, assembly software. So these reads will be assembled into longer sequences. They are called the assembled contigs. Then the next step is to bin the sequences. So we can use here the tetranucleotide frequency of each of these longer sequences, and then we can also use the GC content, which is separate for each of the organisms, and then the coverage, like the depth of the organism which are present. Based on that data, we can reconstruct the different types of the or the different species of the microbial genomes which are present in any environmental sample, in any metagenomic sample. Then we have to further identify whether the genome was complete or it was contaminated. So based on the certain marker genes which are present there, housekeeping genes and then other important genes, we can identify whether the genome is complete or not, or it has contamination. If it shows that marker gene coming from the other species, we can identify the contamination also in that particular genome which we have reconstructed there. So that is the process of the construction of genomes. Then the next step is the annotation of the genome, and then we can also do a lot of stuff like comparative genomic studies, and we can also reconstruct the metabolic pathways there to identify the novel pathways or the novel metabolite product producing pathways we can also identify there. So the take-home message from my presentation is that the multiomics offers a complete view of microbial activity. Microbiomes are highly diverse and central to ecosystem function. Amplicon and shotgun sequencing reveal taxonomic and functional insights. There are various tools available. It is not limited to only these ones, but these are the most popular tools which I have listed in my presentation, like SHINE2 for the Amplicon sequence data analysis, Mother, and R is like it is indispensable. So anyone wants to work in microbial ecology, they have to learn R. If Python they can learn, it is the plus point. But R is very important, at least in the terms of microbial ecology. Then there is ASV and ZOTU constructions or the tables. They provide higher resolutions than the OTUs, which is which is becoming the older term. But the ASVs and ZOTUs are more specialized term, and they are more preferred in the ecology studies. Tools for shotgun sequence analysis which are available there, like Megan is very popular, then Kraken, and then the tools for the annotation there, like the CARD, Comprehensive Antibiotic Resistance Database. Get Encyclopedia of Genes and Genomes, and other databases can be used here for the shotgun sequence analysis. And finally, the MACs are very important to uncover the unculturable microorganisms, where we can identify the diversity of the novel microorganisms. With these, like the basic presentation, I would like to finish my talk here. And if you have questions, please ask me. I will try to answer it.
Thank you so much, Dr. Surendra Vikram sir, for delivering such a highly informative, insightful, and simultaneous section. We are expertise in microbial genomic and bioinformatics has given
Thank you.
the power of computational tools in decoding in microbiome. Your journey and contribution have inspired our audience to be deeper into microbial research with speed and scientific rigor. We now have some time for a question answer session. Participants are requested to type their question in the chat box.
Dr. Vikram, can you hear me?
Yes, sir. I can hear you.
This MAG analysis, what you told, no?
Yes, sir.
Is it only a theoretical or practical also?
This is practical, sir. So I have one published paper in environmental microbiology where we have reconstructed the MAGs and then identified a novel potential for the bacterial rhodopsins, which was never reported from the Antarctic soil. So that was the bacterial rhodopsins. So they use, because that is a very highly oligotrophic environment where the nutrition is devoid. So in that case, what we do, those organisms they have the capability to utilize the energy as the back through the light. So
But
that supports their life.
you do the analysis with the minimum DNA or no? You don't propagate initially. So how you recover the DNA that can be propagated?
Pardon, sir, I don't get your question.
No, you do the metagenomic, right, initially.
Yes,
So you get only the DNA sample for analysis.
exactly.
How you use that DNA to construct a library or bring it into mechanism?
A good question, sir. The thing here, what we do in the metagenomics, we go to that particular slide which I have explained here. This one, this one. So when we extract the DNA from the environmental sample, the
Okay.
genomic library, DNA library is prepared. Right. So there should be at least enough amount of the DNA present. If it is present there, then we can generate a shotgun library here by doing the
So you
enzymatic
go back and
shearing.
then take the original sample, right?
Yes.
You go back to that genomic library and then take the original sample.
No, sir. It is not required. Once we have the short reads here,
Yes.
right? So we can assemble them
Okay.
here.
Okay.
Yeah, so we have the short reads here
Ah.
from particular environmental sample. These short reads can be assembled into longer sequences. They are called the assembled contigs.
Yeah. Then how you assemble that? Because you have the mixture of pull up the different fragments.
Exactly, sir. So there are like metagenome assembly tools or the softwares. They work on particular or dedicated algorithms where they use the coverage as the one factor, and then they align the sequences by construct using the K-mer based approach. So they construct they use the De Bruijn graph, that which is a more mathematical algorithm, which is being implemented in the assembly of these sequences. So there are errors in the assembly still.
Yeah. Yeah. That is what
So I
actually I
will
was
admit
concerned.
that, yes. So there will be errors, but still it is practical in use.
Wonderful. Just another actually query was when we
Yes, sir.
develop alpha or beta diversity, we will measure. We take
Yes, sir.
basically very short sequences, right? Of 16s. Can you hear me?
Exactly. It is up to, sir, yes, so we can take up to 500 base pair by using
Yeah.
forward and reverse primer. So
So
these
you
two
don't
these
think
four reason we sequence most of the time.
there can be a possibly error in the sequence means in that much small sequence?
Definitely. So that is one of the limitations of the amplicon sequencing.
Oh, yeah,
But
okay.
I explained the nanopore and the pack bio, right, sir? So those technologies are like replacing it with the
Yes.
Illumina sequencing technology, because Illumina has the shorter read length. But if we implement the nanopore or the pack bio technology, then we can generate the complete
Longer.
length of the 16S gene, and which
Yeah,
is more informative and more accurate.
yeah, exactly.
Right, sir. So that is the one thing can be done. Yeah.
Very good presentation.
Thank you, sir.
On the similar line, actually, I posted one question in the chat box. After PacBoy and Nanopore, whether any development is there on the sequencing technology? Or is it really needed now?
So the need is, sir, to reduce down the error rates in the nanopore and the pack bio.
Yes.
But SMRT, single molecule real-time sequencing, which is more accurate, so which is in use nowadays.
Yeah, we actually did two of the genome sequencing with Dr. Prabhu Patil in IMTech.
I know, sir. Yeah, very well.
And one of that paper is published. Second one, it is actually under review now.
Right, sir.
Yeah. Thank you. Good, very good presentation.
Thank you.
Sir, one question is, which microbiome are you targeting to study?
For my work, ma'am.
Which microbiome are you targeting to study?
In my research, is that the question? Like which microbiome? So we can target any of the microbiome, from like the human gut microbiome we can select, or suppose we want to study the effect of any drug molecule on the animals. So we just decide our, like we plan our study, and then we just use the animals, and then we collect the feces from the animals, and then we identify what are what is the effect of that particular doses on the different animals, and then how it is changing the microbiome of the particular organism. Or we can also study the different agriculture or the crop soil. So how the microorganisms are changing in different crops, or how the different biofertilizer use of biofertilizer, and then the chemical fertilizer is affecting the microbiome of the particular agriculture soil. So like we can implement it to study any kind of the microbiome from any environment.
Okay, sir. Next question is, what are recent developments in sequence technology than PacBio and nanowire technology?
This is
So
basically discussed already.
yes, this is discussed, ma'am. It is the SMRT can be used also. And I'm looking for the question, ma'am.
How we can use computational approach to predict impact of nanoparticle on microbes?
Exactly, ma'am. So how we can implement it? So like use of nanoparticle can be studied in agriculture soil. We apply the nanoparticle in the agriculture field, and then we study the effect on the microbiome there. Like in the rhizospheric microbiome, we can study the open soil microbiome, how the nanoparticle is affecting, and then how the important flora, like if the yield was enhanced, so which microorganism was affected, which was like increased, which was reduced. So those kind of the studies can be performed by using the nanoparticle based molecules. Like other experiments can also be designed here, but that is one of the kind I explained here. Is it able to list for Leishmania donovani from patients like polyamine pathways somewhat differently in different strains? Uh,
Okay. Sir, next is Megan is free or paid?
uh, I can't hear it, ma'am, properly.
Megan is free or paid?
Yes, ma'am, I explained it there. Megan is free for the community edition. So there are two versions available on the Megan website. So one is the community edition, other one is the ultimate edition. Ultimate edition is paid. We have to pay certain amount of the subscription fee to use the ultimate edition. But the community edition is free. So there are certain advantages with the ultimate edition, that is that it comes with, we can use the command line interface with the ultimate edition, and also we can use the keg annotations, because keg is also a paid database now. It is not freely available to the academicians, so we have to buy the keg database to use it. So in that case, Megan ultimate edition provides us the benefit of using keg database when we buy it, when we purchase the license. But community edition is free of cost. Anyone can download and use it.
Okay. So next is, if I have 16S RNA sequence of already identified bacteria, then can we obtain the whole genome sequence? What tools should be used?
From one bacteria, right? Ma'am?
Sorry?
If it is from one bacteria,
Yes, yes.
yes. So 16S gene is just telling us it is what is its affiliation to the taxonomy. Like if it is for example, we have E. coli there, so we can identify it's an E. coli. Right. But if we want to go further down, what are the metabolic potentials? What are the different genes are present in this particular organism? Then we have to do the genome sequence. So we have to do the shotgun sequence of that particular organism genome. So that is like that is the required part. 16S is telling nothing to us, only the affiliation or the identity of that organism only. And nowadays 16S is not the reliable technique also, Like it is not highly confident technique to say that this is a new species. Nowadays, like if we want to publish a novel species or novel genus or like novel organism, novel bacteria, so we have to perform the genome sequencing study. After that, doing the comparative genomic, then only it is accepted that this is a particular species, new species or not, or new genus or not. So it's important.
Sir, it is able for Leishmania donomani from patient like polyamine pathways somewhat differently in different strain. Kindly address, if possible, HP receptor and pathways analysis.
So, ma'am, this question is like a little bit out of my domain, so I cannot exactly say here. But for Leishmania donomani from patients like polyamine pathways, somewhat differently in different strains, it will be possible. So the thing is, if we have the genome sequence, we can identify those particular locus which are present there, like those particular sequences we can identify in genome, and we can certainly say that whether there are differences or not. But I'm not sure about this until unless I go through more into the research of this particular organism.
Okay, sir, last question is using the whole genome sequence, through what tools can we find the exons for some specific function?
Exons are present in eukaryotic genomes, right? So, whole genome sequence through what tools can we find the exons for some specific function? So that there are different tools available for the annotation of the eukaryotic genomes, like the BUSCO, and then there are different pipelines which are available for the eukaryotic genomes to annotate their functions. So those tools they have implemented there for the exons identification also. So those tools can be used. I'm not very well versed with the name of those tools, but certainly there are tools that can be used. So I'm really sorry about this particular question where the name of the tools are being asked.
Okay,
But there are tools available, yes.
okay, thank you, sir. If any queries arise later, you may connect with us through the Microbiology Society's official communication channels. So, to express our heartfelt gratitude, the Microbiology Society extend a virtual token of appreciation to Dr. Vikram sir for his exceptional presentation and valuable time. Thank you, sir, for guiding us with such clarity and expertise.
Thank you, ma'am. Thank you very much. And thank you, sir. Thank
Yes.
you, Deshmukh, sir. Thank you.
So I would like to gratitude. So our esteemed guest speaker, Dr. Surendra Vikram sir, for sharing his deep expertise with us. I also thank you, Dr. Deshmukh sir, for his leadership, our organizing committee for their dedicated efforts, our technical support team for ensuring a seamless virtual event, and finally, our wonderful audience for joining us and contributing to the success of this program. With this, I formally declare the online lecture concluded. Wishing everyone a pleasant and productive evening. Thank you. Thank you so much.
Thank you, ma'am.